Hey guys, today we're going to talk about part one of chapter 13, disease transmission. And in part two, I'll talk about microbial mechanisms of pathogenesis and epidemiology. But for part one, we're just focusing on the first two bits, disease and transmission. So, first off, what is disease? Well, we define infection as when a contaminating organism evades the body's external defenses and multiplies within the host. Disease is when infections may lead to disease, and it results when normal body functions are altered. Symptoms are subjective characteristics of a disease felt only by the patient. Signs are objective manifestations of the disease observed or measured by others. So, like a fever of 102 degrees, that would be a sign, whereas a symptom might be a headache or a sore throat. Asymptomatic. This is a term for infections that lack symptoms but may still have signs of infection and can be transmitted. There are several stages of infectious disease, and the 5 phases are an incubation period. This is the time between infection and the first symptoms and signs of disease. The prodromal period is a short time, and this is when symptoms are mild, general, and vague, like you just don't feel right. Illness is the severe stage. This is where the signs and symptoms are most evident. Decline comes next. This is a gradual decline of signs and symptoms as body returns to normal function due to either immune response or treatment. And the 5th stage is convalescence. So this is when a patient recovers, they have no signs or symptoms at this point. Patients can be infectious at any stage of the disease, depending on the causative agent. All right, here's a graph of some general patterns of the stages of the infectious disease over time. And the red line indicates the number of microbes or the intensity of signs or symptoms. So when are you most contagious and when do you feel the most sick, right? All right, so the incubation period over here on the left, when you're first exposed, so like right here, at the origin would be when you're first exposed to the pathogen. And then after some time, the prodromal period comes next. This is when you have vague general symptoms, you're just not feeling quite right. And then you move into the illness stage. So this is where you're the most sick, most severe signs and symptoms. As your immune system kicks in or you begin to get proper treatment, you enter the decline phase. So this is when symptoms and signs are less severe. They're declining with time. And then finally, convalescence is when there are no signs or symptoms. Terms used to classify infectious disease. Acute disease, this is probably what we think of most when we picture disease. It's rapid onset. It takes a short period of time, something like the common cold or the flu. Chronic disease develops slowly. It's continual or recurrent, something like hepatitis C or tuberculosis. And then the last one is latent disease. This is when a pathogen remains inactive for a long period of time before becoming active. For example, shingles, right? Shingles is a disease we see in older adults, I want to say like age 60 and up, but it's caused by varicella, or the very same virus that causes chickenpox. So it would be present in an individual for more than 50 years before actually causing shingles. How do we acquire a disease-causing organism? Well, they come from reservoirs of infection. A reservoir is a site where pathogens are maintained as a source of infection. The reservoir may or may not be harmed by the pathogen. And there are 3 general types of reservoirs. A human reservoir. This is the principal living reservoir is a human body. People with signs and symptoms may transmit the disease. And infected individuals who are asymptomatic but infective to others, either before or after the onset of illness. Carriers are individuals that remain asymptomatic and infective for years. This could be someone with tuberculosis, syphilis, or AIDS. They have the disease, but they may have no idea, and they can continue spreading the disease to others. Animal reservoirs are for zoonotic diseases. Diseases naturally spread from animal hosts into humans. And we acquire zoonoses through various routes. So it could be from direct contact with an animal or its waste, something like E. coli 0157H7. You can acquire it through animal bites, like rabies, or even insect bites, like Lyme's disease. Reservoirs can also be non-living. Soil, water, and food can all be reservoirs of infection. Generally, this is because of the presence of microorganisms, often due to contamination by feces or urine. They can be naturally occurring in the environment as well, like tetanus and botulism. Tetanus naturally occurs in the soil. Water can also be a reservoir, especially for something like parasitic eggs like Giardia lamblia or E. coli. Meats and veggies can also be reservoirs for things like E. coli and salmonella. And usually that's because these things are contaminated with feces of an infected animal. Many pathogens cannot survive or remain active for long outside their host. A successful pathogen needs to overcome the innate and adaptive defenses long enough for the pathogen to multiply and exit the host. It's a disadvantage for a pathogen to actually kill the host, since the opportunity to be transmitted may be limited and it loses the source of nutrients. And this idea is called balanced pathogenicity. And actually, we've seen a really good example of this with coronavirus. So COVID-19 is a disease caused by a specific coronavirus infection. But what we noticed at the onset of the pandemic was that COVID infection was very severe and had a high likelihood of death, right? But over time, as we gave the pathogen opportunity to evolve, it developed balanced pathogenicity. A COVID infection today is not typically as lethal as it was in late 2019, early 2020. Now, modern COVID is generally a disease that is more infectious, but causes less severe symptoms and it's less lethal than it was at the onset of the pandemic. All right, movement of a pathogen into a host. How does it get into our bodies in the first place? Portals of entry are sites through which pathogens enter the body. And there's 4 major pathways we'll talk about: the skin, mucous membranes, placenta, and parenteral routes. So for the skin, outer layers of dead skin cells act as a barrier to pathogens. We also have our own natural microbiota that helps prevent pathogens by competitive exclusion. However, some pathogens can enter through openings or cuts. Others burrow into or digest the outer layers of skin, like the scabies might. Mucous membranes are another portal of entry. They line the body cavities that are open to the environment. Things like your lungs and the respiratory tract is the most common site of entry. Entries through the nose, mouth, or even through your eyes. And the gastrointestinal tract can also be a route of entry for some pathogens. They have to be able to survive the acidic pH of the stomach, however. The placenta typically is another potential portal of entry. It typically forms an effective barrier to pathogens. However, some pathogens can cross the placenta and infect a developing fetus. These infections can cause spontaneous abortion, birth defects, or premature birth. And a good example of this is lunch meat. Lunch meat can contain listeria, a pathogen causing listeria. So if you're pregnant or over 50 and at a higher risk for listeria, it's recommended that you heat your deli meat, your cold cuts, up to 165 degrees or steaming. The last portal of entry is the parenteral route. And it's not truly a portal of entry. It's a way that other portals can be bypassed. So it's when pathogens are mechanically deposited directly into tissues beneath the skin or mucous membranes. So it could be from a hypodermic needle or a thorn or stepping on a nail. Those would be parenteral routes of entry. Let's talk now about modes of infectious disease transmission. We know how it gets into our bodies, but how does it get from person to person? Transmission is from a reservoir or portal of exit to another host portal of entry. And there are three groups of transmission. There's contact transmission. This could be direct, indirect, or via droplets. There's vehicle transmission, like airborne, waterborne, foodborne, where something that we take into our bodies normally carries a pathogen. And there's also vector transmission. It could be biological or mechanical, but vector transmission is when an organism or mechanical object deposits a pathogen into a new host. So direct contact is a type of contact transmission, and it's when one person physically touches another. It could be pathogens spread by handshakes, sexual intercourse, or a fecal oral route. There's indirect contact, and this is when pathogens are transferred by an inanimate object called a fomite. So it could be clothing, doorknobs, glasses, and eating utensils. For example, scabies is often transmitted by bedding that contains the scabies mite. All right, another type of contact transmission is by droplets. Transmission of a pathogen through inhalation of respiratory droplets. Close proximity is usually necessary. They can be transferred by coughing or sneezing, and some examples of this are the cold, influenza, and coronavirus. All right, let's talk now about vehicle transmission. Airborne transmission is when pathogens are suspended in the air and they travel through the air to another host. It's very difficult to control airborne transmission. These droplets can remain suspended for long periods of time, and they leave a host by coughing or sneezing. And some examples of this are tuberculosis and measles, which we know are highly contagious. All right, let's talk about food as a vehicle for transmission. It usually transmits digestive tract diseases, and contamination can occur in a variety of different ways. Animals can harbor pathogens like salmonella in chicken or E. coli 0157H7 in ground beef. It can also inadvertently be added during food preparation. If cooks don't wash their hands properly, some of their own Staphylococcus aureus can enter the food. There could be cross-contamination, like a cutting board used on meat and vegetables for your salad. If I cut up raw chicken breast to cook and then I use the same knife and cutting board to cut up carrots for a salad, that could contaminate your salad with salmonella. It can also happen in meat and vegetable processing facilities. And another way is also by improper butchering or processing practices. All right, let's talk for a moment about foodborne intoxication. This is the ingestion of an exotoxin produced by an organism growing in a food product. So, a toxin produced by a microbe growing in food. The toxin causes the illness, not the living microbe itself. The organism is killed by heat, so even if you cook your food thoroughly, you may still get sick from the toxin. And so often these toxins are heat stable. So some examples of this are Staphylococcus aureus and also Clostridium botulinum. Clostridium botulinum is what causes botulism. And this is something to keep in mind if you can foods at home. You need to be leery of this and make sure that your fruits and veggies you're canning are all very clean, because this is a normally occurring soil microbe. It produces a toxin that's heat stable. So even if your canning effectively kills this organism, the exotoxin it produces could still be there in your canned foods. All right, foodborne infection. This requires consumption of living organisms. Some examples are like consuming undercooked hamburger or chicken. Symptoms don't usually appear for at least a day later. And diarrhea is kind of the classic one that comes to mind, right? Thorough cooking before consumption will kill most foodborne pathogens like E. coli 0157H7 and Salmonella. All right, let's talk about water now as a vehicle of transmission. This can often involve very large numbers of people. For example, in 1993, Cryptosporidium parvum, an intestinal parasite, infected about 400,000 people in Milwaukee from a municipal water source. The infectious dose is less than 10 organisms. It takes very little Cryptosporidium to make people very sick. And when I say very sick, I'm talking watery diarrhea on the order of 10 to 15 liters a day. That's a ridiculous amount of volume. All right, vector transmission. So vectors could be biological, they could be living things, or it could be mechanical. So a biological vector is an organism that transmits and serves as a host. The vector is involved in the life cycle of a pathogen. For example, malaria. Part of the life cycle of the protozoan Plasmodium occurs inside an Anopheles mosquito. A mechanical vector transmits disease causing a disease-causing organism through mechanical contact. An example for this is trachoma or blindness, and it's when Chlamydia trachomatis is carried on the feet of a fly from an infected person's eye to a new host. All right, how is it that pathogens move out of a reservoir? We term this a portal of exit. Pathogens leave hosts through portals of exit, and many portals of exit are the same as the portals of entry. Pathogens often leave hosts in material the body secretes or excretes, and human activities promote the transmission of the organism. So some of these possible portals of exit are their blood, skin, mouth, nose, ears, eyes, and different bodily secretions. All right, so that wraps up part 1 of chapter 13.